Hi, my name is Kobe Hayes, and this is my discussion over oxidative phosphorylation. In the beginning, we have glycolysis, which is a series of reactions that extract energy from glucose by splitting into 3 carbon molecules called pyruvate. That is the full, right here you can see the full transformation of glycolysis from glucose to pyruvate. Each pyruvate from glycolysis goes into the mitochondrial matrix. There it is converted into a 2-carbon molecule bound to coenzyme A, which is acetyl-CoA, which is then turned into the citric acid cycle. The acetyl-CoA goes through a cycle of reactions, regenerating the 4-carbon molecules, starting up ATP, NADH, and FADH that are produced and then releasing carbon dioxide. We then have the Krebs cycle, which is also, one of the important parts of oxidative phosphorylation. It is where energy is stored in pyruvate, which is transferred to NADH and FADH2, and ATP is then produced. The end result through oxidative phosphorylation is the electron transport chain. The role of the electron transport chain is to basically produce ATP molecules, but also is a collection of proteins found on the inner membrane of the mitochondria. NADH is releases the hydrogen ions and electrons in the transport chain. The mitochondria, which is obviously the powerhouse of the cell, is important in cellular respiration. In cellular respiration, mitochondria is important because it is the main production of ATP. It uses chemical energy found in glucose and other nutrients. It's also important in producing other atoms that are important cofactors of many enzymes. Chemiosmosis is the actual production of ATP in cellular respiration. Chemiosmosis involves the pumping of protons through special channels in the membranes of mitochondria from the inner and outer compartments. It is the movement of ions, especially protons, across the membrane, resulting in an electrochemical gradient that drives ATP synthesis. In cellular respiration, there is also a protein gradient. The protein gradient is a higher concentration of protons, which is hydrogen ions, on one side of the membrane than the other. So for instance, you can see how hydrogen is pumped through to the inner membrane space and also the inner mitochondrial membrane through a proton pump, which therefore produces more hydrogen ions on one side of the membrane than on the other. To conclude, the electron transport chain and chemimeiosis make up oxidative phosphorylation. In these steps, the delivery of electrons by NADH and FAD2 is occurring. Electron transfer and protons are being pumped. There's a splitting of oxygen to form H2O, and the gradient-driven synthesis of ATP is also being showed. Thank you for listening to my discussion, and I hope I was a help to you. Thank you.